and welcome to this video for higher biology. In this one we're going to be looking at gene mutations. So up until now we've just been looking at genome structure and the fact that the genome is made up of both coding and non-coding regions. And what we're going to look at today is mutations in the genome, what happens when these occur on a single gene, and then we're going to go on to look at chromosome mutations, so what happens when a mutation occurs on a whole entire chromosome. So what you need to be able to do is explain what we mean by the term mutation. You need to be able to state the effect of a mutation on the protein that's produced. You need to be able to describe and give examples of the three types of gene mutation, and you need to be able to explain the possible consequences of each type of mutation. First of all, what is a mutation? Well, a mutation is a change in the DNA that can result in no protein or an altered protein being produced. And as we said, mutations can occur in a single gene and therefore affect one protein, or they could affect part of or a whole chromosome and consequently several proteins. Mutations themselves are quite rare. In humans, it's estimated that there's about one mutation for every 15 to 30 million base pairs, so really quite rare. That rate of mutation is going to be different from organism to organism, so different species will have a different rate of mutation, and also some genes are more likely to be mutated or not. So clearly genes which are vital for survival are less likely to mutate. If they do, that mutation wouldn't be passed on, as that individual is less likely to survive. Other mutations that might be a little bit more frequent might be those which aren't necessarily going to have an impact immediately on the viability of that organism, on whether or not it's going to survive, even surviving birth, to the point at which it might survive adulthood in order to be able to reproduce and pass on that mutation. So if we think back to what we've been looking at already in terms of protein synthesis, so how does a mutation actually affect a protein? If we consider the order of bases in our DNA sequence, that is going to be transcribed and will produce an mRNA sequence. That sequence of mRNA is going to be determining the order and the specifics of what amino acids are going to make up the protein. So once the exons and introns are spliced, that mature mRNA transcript is going to be taken to the ribosome, where it will be translated into our protein. And with the order of each of those codons, so that group of 3 bases on our mRNA, is going to code for a specific amino acid, which will be put together in this long polypeptide chain forming our protein. So the order of bases on DNA determines the order of bases in the mRNA, and the order of bases in mRNA, when it gets to translation, will determine which amino acid it is that we're including in the protein. So when we have a mutation occurring in just one section of a gene, then the overall result for the protein itself is it may have a changed shape as a result of a different amino acid being included within that protein, or it might not be produced at all. So in this case you can see that this one here, we've swapped this G for a C. The rest of the sequence is absolutely the same. However, this one's making a much larger protein and this one's making a much smaller protein. So again, if this one's going to be vital for survival, then when this embryo develops, it's less likely to actually continue to develop and may not be viable with life. If we have a less serious protein or a less consequential protein being used, then this individual may continue just to express a small part of the protein. This may result in them, for example, if this was an enzyme, they may not be able to break down a particular molecule particularly well. So as I said, we're going to just be looking at single gene mutations today. So mutations which only affect one nucleotide in one gene. They may also be referred to as point mutations. If you're looking at any older textbooks or older past paper questions, you may see them referred to as point mutations as well as single gene mutations. And really, a point mutation just means it's occurring at one point across the DNA sequence. So they may be a substitution, a deletion, or an insertion of a single base. And it's important to realise that this might occur in the protein coding sequence, so a section that will affect a protein, or it might actually affect the regulatory sequences which control expression of a gene. So as we just looked at when we were looking at genome structure, the section of DNA which actually doesn't code for a protein particularly, but is still used in regulating gene expression. If that's mutated, then obviously that gene may not actually be expressed at all. So although one single point mutation is only going to affect one small part of the DNA, it could actually have a larger impact in preventing a gene from being expressed and therefore a protein produced entirely. So we're going to first of all look at substitutions and the effects of substitutions before we have a quick look at insertions and deletion mutations. So a substitution mutation is a bit like it sounds. If this is our original DNA strand and codes our mRNA, which gives us our amino acid order. If we have a substitution mutation here, our original DNA strand, we have a T has been substituted for an A. So literally they've just been swapped over. The number of bases included is the same. We haven't removed any, we haven't added in any. We've just literally swapped a T for an A. And as a result, we have an ever so slightly different mRNA molecule coded. So we've got a U in here instead of an A. And this has resulted in a different amino acid being included in our final polypeptide chain. So that would be what a substitution mutation would do. It would swap one base for another base. Now, the implications of this are very much determined by which base is swapped for which base and what that does, the effect that has on the swapped amino acid. And there's a few different options that we might come across. So in this case, we might see a silent mutation. In that case, what we're seeing up here is if we look at our original DNA molecule, we've got our CCG, which is the same as it is down here, CCG, and that is going to code from our mRNA, it's going to code to proline and code to proline, so that's fine. If we look at this one, we've got GAG, however in our mutated version we have GAA, but actually as it turns out they both code for glutamic acid, so there is no actual observed change to this mutation. All we've had is we've substituted this G for an A, but it's resulted in the same amino acid being included within the polypeptide chain. And that's what we refer to as a silent mutation, because you wouldn't notice it, the organism wouldn't have any particular changes to it. We've got exactly the same protein as we would have had originally. A substitution may also result in what we refer to as a missense mutation. And in this case, we've got a similarly substituted base. We've got our CCG in our mRNA coding for proline. We've got a CCG in our mRNA coding for proline. And in our swapped over one, we've got GAG is our original, but we've got GUG. And in this case, that's where we've swapped glutamic acid for valine. And the consequence for that is that we've wound up with a different amino acid. So in this case, the original protein, this one here, would actually give you normal hemoglobin. And that's the protein that binds to oxygen in our red blood cells and makes our red blood cells red. However, in the substituted version, just that one change from that A to that U and that inclusion of valine in our protein instead has given us the mutated protein, which is hemoglobin S, and that's the one that actually causes sickle cell anemia and causes the red blood cells to be shaped wrongly and therefore not carry as much oxygen. We also have what we refer to as a nonsense mutation. Again, this is where we have a substituted base, so it's a substitution mutation, and the consequence is that it's a nonsense mutation. So in this case, we've got our substitutions at the beginning of the codon. We've got this G swapped for a U, and so instead of GAG giving us glutamic acid, instead we've got UAG, and that's given us a stop codon. As a result, that's going to mean we've got a much shorter polypeptide chain than normal. So that's either going to form a protein that doesn't function at all or form a slightly different protein. So that's going to have a big and significant consequence to the organism if we have a nonsense mutation. So substitution can result in it being silent, it can be missense, or it can be nonsense. And silent is where we have the same amino acid produced anyway. Missense is where we have a different amino acid produced and it might affect the shape or style of the protein. And nonsense is when we wind up with a stop codon, therefore we wind up with a shorter or truncated protein. And that's all the result, those three types are all the result of a substitution, one base being swapped for another. You can also have some mutations which result in us having a different number of bases. So again, if this is our original DNA strand, coding for mRNA, coding for our amino acid polypeptide chain, in this case, we have an insertion mutation. So an extra nucleotide has been inserted into our DNA strand. So if you look at our original, we've had AGA as our first codon, matching up with UCU in our mRNA coding for the serine. Over on this side, we've had AGA, UCU, that's coded for our serine molecule. However, our next one, we should have had GTC. However, we've now got GGTC. So we've got an extra G in there, and that's given us a different codon, which has given us a different amino acid. We can also have the converse occur. So in this case, same original strand, however this time round we've removed the T in the middle, so we've lost this T, has come out from our original DNA strand, it's been deleted. And as a result, you can see we still get our original serine, but then instead of glutamine we've got arginine included instead. Now those of you who've been paying close attention will have noticed that actually it's not just arginine that's changed, we also have a different one at the other end, and it was the same with the insertion mutation. And this is because both insertion and deletion mutations result in what we call a frame shift. And that means the frame of 3 that the codons are read in has been changed. So it's not just the fact that it's affected this one codon here. If we've taken one out, if we've removed it and made it shorter, we've shuffled all of these up the way. They've all gone that way, so we're missing one. So not only has this codon changed, but so has this one, and all of the ones coming after it. So a frameshift mutation can be quite significant. So anything that inserts or deletes a nucleotide is not only going to affect the amino acid that that particular codon was coding for, but it's also going to affect all of the amino acids which come after it. So insertion and deletions result in what we refer to as this frame shift mutation. We also have what we call splice site mutations. And this is when the DNA that's being mutated, the mutation actually occurs in an area which affects how that mature mRNA is put together. So we might wind up, if this is our original DNA, and we've got exons and introns, removing the introns, putting them together we're putting our exons together to form our mature mRNA transcript. If we have a mutation in this sequence here, we might wind up with the intron, this bit here, is going to be included accidentally into our mature transcript. Or if we have another mutation somewhere else, it might mean that that's read as an intron instead of an exon, and it would be excluded from the mature transcript. So a splice site mutation might be caused by a substitution, which essentially means that the introns and exons get a bit mixed up, and we either include an intron in the mature transcript or exclude an exon. So thinking about gene mutations, you should know that a mutation is a change in the DNA that results in either no protein or an altered protein being produced. The effect of a mutation on the protein produced could be to change its shape or to stop it being produced entirely. That the 3 types of gene mutation are substitution, insertion, and deletion. The consequences of each type of mutation: if it's a substitution mutation, it might be silent, so the amino acid stays the same. It might be a missense, the amino acid is swapped for a different one. Or it might be nonsense, where we wind up with a stop codon instead of the correct amino acid. The consequences of insertion deletion mutations are that we have frame shifts, so every amino acid after the insertion or the deletion of the nucleotide results in a change to the rest of the amino acids following on from that. As ever, feel free to ask any questions at all as we go over this in class, and we'll be making sure to get a lot of practice on this later on.